Every creature is made from a recipe that comes from its DNA, spelled out in chemicals A's and C's and T's and G's inside the famous double helix. So how do you get from DNA to become a real creature? Well, let's take one of those fantastic voyages and we'll show you. We're going to find DNA and, well, we'll make it a typical cell, so we're going to have to fly in and then go off to the nucleus of the cell, which will make a beautiful castle, the headquarters. And there's the DNA, the master code inside the nucleus. DNA never leaves the nucleus.
You ever meet one of those mean librarians, you know?
Yes.
Special reserve section?
The ones that go, pow.
Right. You can take the thing, you can copy it, but you can't take the book, because somebody else might need it.
So if DNA is locked in the nucleus, how do we get the information out to build our creature? Well, that scribe, copying recipes out of the cookbook and throwing them out the window, out to the cytoplasm C that makes up most of the cell, all those recipes floating through the air, they are RNA. And to finish up in that C, you see hundreds of thousands as well. We've made them into little guys with chef hats.
Those would be ribosomes, and in your world, there are chefs who are using the recipes that are written in the RNA.
And whenever a recipe lands on a chef, whatever it is, he cooks it.
Whatever it is, he cooks it.
And each recipe is for a protein. Proteins build cells, bone cells, brain cells, all cells. So all these chefs are basically building you. You are made of proteins. And the discovery of RNAi was an accident. It was a puzzle that appeared in a petunia.
Maybe this.
In 1986, geneticist Rich Jorgensen was working at a biotech startup company in California. He was asked to create a spectacularly dazzling flower. And so they decided to create a very, very, very purple petunia. Rich knew which gene produced purple. He knew how to sneak an extra copy of that gene into the plant's DNA, the master text, to be copied by that monk-like scribe.
It will be transcribed by the monk, the same as any other gene. And he'll throw the transcript out the window into the cytoplasm, where the chef will be able to pick it up and use it.
Rich thought that if he added more purple recipes, he'd get a purpler petunia. So he did it, and he waited. And what happened?
We produced, instead, white flowers.
White flowers?
Complete opposite of what we had expected. Our initial reaction was that something must have been wrong with the gene that we had engineered to introduce to the plant.
A mistake?
A mistake. So we checked everything out, and there were no mistakes that we could find.
The petunia was a big puzzle. Nobody understood why, when you add an extra gene for purple, you should not get more purple, but less purple. It took a decade of brilliant scientists working on petunias and fruit flies and worms and other organisms to finally work out what was going on.
Quite by accident, Rich had discovered a secret inside living cells. Cells from time immemorial have had a mortal enemy called the virus. So let's imagine that the virus is a pirate ship. It lands. It then sends invaders inside the cell to shower recipes down to those cooks.
The sole purpose of that virus is to make additional copies of itself, to the point that the entire cell is filled up with this. And the cell explodes, releasing these viruses to go and then infect whatever other cells they can find.
So the theory is that long ago, cells developed a secret defense system which we will call the cop. What the cop does is when viruses invade and create showers of murderous recipes, the cop looks and thinks, some of these have a very fishy shape. It's a chemical difference which comes down to some of the viral recipes are 2 pages instead of 1, and one side is a mirror image of the other. But the point is, to the cop, there's something not right about this shape. So when they see it in that shape,
They say virus.
And
They
go,
say,
uh-oh. And the cop destroys the recipe.
Cells were able to tell that something was very funny when they saw mirror image messages and start not just destroying the messages, but destroying anything that looked like that message. They'd worked out this whole defense system against viral RNA, and we then accidentally stumbled into using it.
The question, remember, was when Rich tried to make his petunia more purple, why did it turn white? Well, the answer, it turns out, was that Rich, by accident, discovered the cop. When Rich invaded the petunia cell and inserted his make more purple instructions, he didn't know it, but his purple instructions happened to have that suspicious viral shape. So when the cops saw the recipe, the cop thought, virus, and destroyed every recipe for purple in the cell. If there are no recipes for purple, the chefs don't cook purple.
And because there's no purple pigment produced, the flowers will be white.
And that's how Rich and his petunias helped discover what we now call RNAi.
RNAi means RNA interference, because the cop is interfering with RNA messages, with the recipes in the cell.
I want to share with you my experience working with transgenic plants, particularly the problems that I had because of gene silencing. I did my PhD in Argentina, working in wheat with better nutritional and bread making quality, but I had problems with gene silencing that almost prevent me from getting my PhD. So here is the short version of the story. I work on the development of transgenic wheat with better bread making quality at the CEFOBI Institute, that is the Center for Photosynthetic and Biochemical Studies at the School of Pharmacy and Biochemistry, National University of Rosario in Argentina. The idea was that to use the gene gun to deliver the plasmids containing genes for better bread making quality, and that DNA was coating gold bullets. So the gene gun doesn't look like this, but look more like this. So using these gold bullets that were coated with a plasmid containing the gene to improve bread making quality, we bombard some particles. We use microparticles of gold to bombard something called cales. So are indifferentiated plant cells that are used as a material to be transformed. And then those transgenic callus are put into a media that has hormones that are going to regenerate a full plant. So briefly, these are the steps to develop a transgenic wheat. The first thing is to work in vitro, cell culture that is with plant cells, and then do the selection and analysis of those transgenic plants. And finally, after selecting the best transgenic plants, that are the ones that express and produce a higher level of protein, then we obtain the flour from the wheat and use that flour to make a kind of dough, and then we measure the quality of the dough using an apparatus that looks like this, that is the mixograph. And then the next step will be to make bread. So the subject of my PhD thesis was the improving bread making quality of wheat using recombinant DNA technology. So the elasticity or dough strength is extremely important to obtain a wheat for good bread making quality. So strong doughs are considered excellent for bread and pastas. And originally, when you separate proteins using shell electrophoresis, you see different bands, and these are proteins that are known as glutenins, and these are the proteins known as gliadins. These are the proteins that the celiacs has problems. But glutenins are associated with bread making quality. So the higher the number of these bands, the higher the bread making quality. It's not just the number, it's also the type of proteins. There are a family of proteins, and some type of proteins are associated with better bread making quality than others. So our aim was to introduce another band that we expect to find here, and that increasing the number of glutenin subunits, we will increase bread making quality. So that's why we introduce a plasmid to obtain an extra band here. But what happened is that instead of obtaining an extra band, we have a whole gene silencing of all the glutenins. So this was horrible, not only because it was a result that was very unexpected, I didn't have a clue about what was gene silencing at the time. My thesis director didn't have a clue either, and he thought I did something wrong, and he strongly recommended that I just trash those seeds. But I never give up. I decided that these results were quite interesting, and I didn't follow his advice, and I continued working with these seeds, and I consider this plant particularly important. So another thing I did, besides not giving up, was to read more, trying to find out what you could do with a wheat that has a very weak dog. But the main problem I had at that time was that I had to propose a different title for my thesis that would say worsening of bread-making quality of wheat by recombinant DNA technology. And of course, nobody will be interested in seeing how I will make the bread-making quality worse. But what I found is that weak dough can be used to make cakes and crackers. So that could be an application of my wheat that has that gene silencing. So fortunately, we continue obtaining other wheat lines, and I finally obtained one that has the expected band that is showing here. So this wheat really has an improved in the bread making quality, and that's why I was able to go back to the original title of my thesis, and that's why I was able to finish my thesis. But actually, those results with gene silencing make it much more interesting. And then I published my first paper that was about silencing of high molecular weight glutenins in transgenic wheat expressing extra high molecular weight subunits. And this is, of all my more than 30 papers, is the paper that has the highest number of citations, more than 200 citations. This is an example of how you should not give up. Even when things looks very bad, you can find a way out by reading, by persevering, by trying again, by trying to find a new option. And in science, the results that are the most unexpected are the results that ended up being most interesting. Then I went to Spain to try to analyze the dough of these different lines of transgenic wheat, some with partial gene silencing, others with complete gene silencing, and other ones expressing this extra subunit and have really high bread making quality. And the results are shown in this paper that we did in collaboration with the Spaniards. And then many years later, when I came to the US and I started working on developing a transgenic tomato, with the containing a plague, bubonic and pneumonic plague vaccine. This is the work I did at ASU. We have again problems with gene silencing, but we use a strategy to reverse gene silencing. So the strategy we copied from a virus that is able to escape from these gene silencing in plants. So when we try to express simultaneously the F1B gene, that was what we were trying to express to obtain the vaccine for plague, together with the P19 gene that was from a virus that was able to reverse gene silencing, we obtain a very high level of the F1B protein, that is the one that is used for the prevention of pneumonic and bubonic plague. So this is how many years later I use all my experience with gene silencing to find a way to revert it, and in turn obtain plants that express very high levels of the transgene. So my results appear not only in scientific papers, but also in Argentinian newspaper. In 2001, for example, here say an Argentinian obtained transgenic wheat for the first time in the country. Then, unfortunately, another article say the odyssey of doing science in Argentina. That's why I had to end up coming to the US, because Argentina in 2001 was in the worst economical moment of our history, so it was very hard to find a job, and that's why I decided to come to do my postdoc at ASU. So here another paper also reflecting the problems in Argentina to keep the scientists because of the low salary or how hard it is to find jobs. is the use of this technique to treat disease by knocking down specific unwanted or harmful genes that are causing disease. So how we do that? What is the mechanism? How we do to use RNA therapy? The only thing that we had to do is to synthesize double-stranded RNA. Remember that RNA in the cell is normally a single-strand molecule. But the double-strand RNA that is complementary to the sequence of the target gene, the gene that we want to knock down, is going to be introduced in the cells and is going to turn off or silence the target gene. So in other words, the target gene is going to be inactivated and is not going to be transcribed or translated, and therefore is not going to produce the final product, that is the protein. So using these strategies, different kind of disease can be treated, and some of them are currently during clinical trials. So some of these diseases are genetic disease, such as macular degeneration, that is the leading cause of lost vision in the US, and Huntington disease, that is a genetic disease in which the neurons are degenerate very fast. Then also RNAi therapy can be used to treat different infectious diseases, such as respiratory syncytial virus, herpes simple virus, hepatitis A, B, and C, and HIV. All these are current or past clinical trials, and most of them has been successful. RNAi therapy can also be used to treat cancers such as lymphoma, melanoma, solid tumors, etc. So now we are going to see a clinical case of a woman with macular degeneration and how RNA therapy helped her to at least partially recover her vision.
Years ago, Marty enjoyed doing lots of things, but then she began losing her sight.
Couldn't see, and I'd probably get the peppers in with the zucchinis, and there'll be big problems then.
So she went to her doctor, who told her,
You have macular degeneration.
a degenerative disease caused by too many blood vessels growing in the eye, underneath the retina.
As these blood vessels grow, they leak out fluid and blood in the center of her vision, and it's as if you're looking through a very dirty windshield, essentially.
I went home, I was just devastated.
But Marty volunteered to be a candidate for RNAi therapy, something so new she's kind of a pioneer.
She was probably one of the first to get it for any disease whatsoever, and specifically for macular degeneration.
You don't hear much about it, but RNAi is a really big deal. When scientists realized that every plant and animal cell has RNAi, a way to turn off the recipes, turn off genes, they thought, maybe we can use these cops to work for us. The reason Marty has so many blood vessels growing in her eye, clouding her vision, is there's probably a mistake in her DNA, in a gene that produces too many recipes that say, make more blood vessels. So the chefs cook up proteins for more, and she ends up with too many blood vessels. Her doctor wants her to have fewer blood vessels. But how do you get the chefs to make fewer blood vessels?
It's pretty easy. You want to shut down a gene? Put in a copy of the gene with its mirror image. Signals the cell, better shut this thing down.
We inserted the needle after numbing her up. Down below
So the
white part
doctors
of the eye,
literally
we
injected
inserted the
RNA
needle
recipes
after
into
numbing.
Marty's eyes that said, make more blood vessels. But they made those recipes look dangerous, like viral recipes, hoping the cop in Marty's cells would leap to it and destroy lots of recipes for more blood vessels, leaving Marty with fewer blood vessels. They wanted the cop to turn off Marty's disease. Did it work?
Marty's vision has improved. It's a very promising result.
So apparently they did trick the cop in Marty's cells to reduce vein production, although not completely.
I see the yellow. Inside is just a little cloudy, but I can see it.
There's a lot of questions still that need to be answered.
Can we deliver recipes to the right cells?
Lovely.
Does the treatment last?
That's beautiful.
All these are big questions. Still, in mice, RNAi has been effective with Huntington's disease, Lou Gehrig's disease, hepatitis, breast cancer. So if we ever work this out in humans...
Any sort of disease that you can imagine becomes fair game.
So any questions you might have about my work, just about in general, about gene silencing and RNAi therapy, I'd be very happy to answer. Thank you.